Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
1 in 8 men in the United States. That's how many will hear the words, "You have prostate cancer," during their lifetime. It's a huge number. And even with all the advancements we've made, it's still the second leading cause of cancer death in American men.
It really is.
And one of the biggest challenges, figuring out what to do when the treatments stop working, when the cancer becomes resistant. That's what we're diving deep into today.
Yeah, that's right.
Brand new research, hot off the press. They've uncovered a key mechanism driving this resistance in prostate cancer. And the really exciting part, it could point us towards new ways to fight back, potentially overcoming this resistance.
Exactly. And it's something that researchers and clinicians have been grappling with for a while.
Absolutely. We've got two main sources for our deep dive today. A health news article that breaks down the findings in simpler terms,
Right.
and the original scientific paper published in the journal Oncogene.
The nitty-gritty details.
So let's break it down. What did these researchers find? What does it mean for potential new treatments? And how complex is this stuff, really?
That's a great place to start.
Okay, so to really get this new research, we need to kind of start with prostate cancer treatment 101.
Okay.
You mentioned earlier that researchers have gotten better at treating prostate cancer by targeting something called the androgen receptor.
Yeah.
Can you remind us what that is, and why is it such a big deal in treating this particular cancer?
Sure. So think of the androgen receptor as like a doorway on a prostate cancer cell.
Okay.
Specific hormones like testosterone act like keys that fit into this doorway.
Okay.
When they do, they send a signal inside the cell that tells it, hey, it's time to grow and multiply.
Got it.
Prostate cancer cells are really dependent on these signals to grow.
I see.
So that's why the androgen receptor is such a prime target for so many treatments.
That makes sense.
Block that doorway, and you can potentially stop the cancer in its tracks.
So that's where things like androgen deprivation therapy and androgen receptor signaling inhibitors come into play.
Yeah, exactly.
What do those treatments actually do?
All right, so androgen deprivation therapy, that's all about lowering the amount of those growth-promoting hormones in the body. Less fuel for the cancer cells.
Right.
Then you have androgen receptor signaling inhibitors.
Mm-hmm.
Those are drugs specifically designed to, like I said, block that doorway.
Okay.
So even if the hormones are around, they can't get in to deliver their growth signal.
They can't turn that key.
Exactly. And these treatments have actually been pretty effective for a lot of men, especially when the cancer is caught early.
But as we said at the beginning, prostate cancer can be tricky.
Oh, yeah?
It often finds ways to keep growing, even when we use these therapies.
It does. It's a bit like a really determined weed in your garden. You pull it out, and it finds a way to sprout back up.
So how does it do that? How does it become resistant?
So imagine this. Over time, some of the cancer cells, they change.
Okay.
And these changes, they allow the cancer cells to either not need those hormone signals anymore or to find totally different ways to grow.
So they figure out a workaround.
Exactly. And this is what we mean by resistance. It's like the cancer is adapting, evolving to survive.
So that leads to what's called castrate-resistant prostate cancer.
Precisely. When the cancer keeps growing, even when those hormone levels are really low or the androgen receptor is blocked.
And I think you mentioned that scientists developed even stronger drugs like enzalutamide to try to combat this.
Yeah, they're designed to be even more effective at blocking that androgen receptor.
Okay. But even those aren't a sure thing, right?
Sadly, no. They work for a lot of men, but for many, the cancer eventually figures out how to escape even these more potent drugs. And that's really the core issue that this new research was trying to address.
So let's talk about this new research. Who are the brains behind this work?
This research came out of the Texas Tech University Health Sciences Center.
Okay.
Leading the charge were Dr. Surimas Nadana and Dr. Manisha Tripathi from their Department of Cell Biology and Biochemistry.
Mm-hmm.
And a shout out to Sayanika Duda, a graduate student who really drove the study forward.
Oh, that's great.
They also collaborated with researchers from Rutgers School of Health Professions.
Now, what were these researchers actually trying to find out? What was their big question?
Okay, so their main goal, they really wanted to understand the exact steps, the molecular processes that drive advanced prostate cancer to just keep going,
Okay.
and especially how it becomes resistant to those androgen receptor blocking drugs like we talked about.
So they're trying to figure out the how of resistance.
Exactly. And ultimately, if they could understand how it works, maybe they could find a way to stop it.
Okay. Now, Their research zeroed in on a specific protein called TBX2. What is this protein and why did they focus on it?
Well, the Nandana lab has been studying TBX2 for a while.
Okay.
And TBX2, think of it like a master switch. It's called a transcription factor.
Okay.
It controls the activity of a bunch of other genes in the cell.
So it's like a control panel with a lot of switches.
Yeah, that's a great way to put it. It can essentially flip other genes on or off. Now, previous research had shown that TBX2 is often found in higher amounts in castrate-resistant prostate cancer.
Interesting.
So that was a clue, maybe a red flag, that TBX2 could be involved in the cancer becoming more aggressive and resistant to treatment.
So they thought TBX2 might be up to no good.
Exactly. They wanted to figure out exactly how TBX2 was contributing to this resistance, particularly to enzalutamide.
The enzalutamide. Okay.
Yeah.
So how did they actually figure this out? Like, what did they do in the lab?
Well, they did some really elegant work, mostly using human prostate cancer cell models.
Okay.
Some of these cells were already resistant to treatment.
Okay.
And one of their key tricks was to actually manipulate the TBX2 protein in these cells.
So they're like puppet masters.
Kind of. They figured out ways to either lower the amount of TBX2, increase it, or even completely block its function.
Interesting. So they were like turning TBX2 up, turning it down, turning it off,
Yeah.
and seeing what happened to the cancer cells.
That's it. And to really see the impact of these TBX2 changes, they used something called ribonucleic acid sequencing analysis.
Okay, break that down for me.
So imagine being able to see the activity of all the genes in the cell at once. That's basically what RNA sequencing does. It's like taking a snapshot of what all the genes are up to.
Oh, I see.
So they could see how messing with TBX2 changed the activity of all these other genes. And then to confirm what they were seeing, they used some other techniques like quantitative real-time polymerase chain reaction and Western blot analysis.
All right, I know those are important, but honestly, they sound a bit intimidating. Can you demystify them a little?
Sure. Those are just ways to double-check and measure the exact amounts of RNA and protein in the cells.
So they're like molecular measuring sticks?
Exactly. They let the researchers zoom in on specific genes and proteins of interest and confirm that what they saw in the RNA sequencing was actually happening at the protein level.
So they went from this big picture view with the RNA sequencing to a more focused, close-up look with these other methods.
Precisely. They wanted to be really sure about their findings.
And didn't they go even deeper, like looking at how TBX2 interacts with the actual DNA, the genetic material?
Oh, they did. They used a technique called chromatin immunoprecipitation assays.
And in plain English?
Basically, this technique allows them to see if the TBX2 protein is physically attaching itself to specific parts of the DNA that make up those genes.
Like the gene for the androgen receptor.
Yes, exactly. And they also looked at another receptor, the glucocorticoid receptor, which we'll talk about more in a bit.
Okay, so that helps them figure out if TBX2 is directly controlling the activity of these key genes.
Exactly. And to be extra sure, they even made tiny specific changes to the DNA to pinpoint the exact spots where TBX2 was binding.
So they're like, TBX2, we see you.
Pretty much. And they confirmed that this binding was absolutely necessary for TBX2 to have its effects.
So it's like they were mapping out TBX2's control panel.
A great analogy. And it gets even more interesting. They also investigated whether TBX2 was teaming up with other proteins inside the cells. They used co-immunoprecipitation assays for that.
Teamwork in the world of proteins.
Always. Proteins rarely work alone. They often interact and form complexes, and these interactions can be crucial for their function.
Like a molecular dance party.
You got it.
Okay. And to make sure all of this wasn't just happening in their lab dishes, they also looked at data from real patients, right?
They did. They wanted to make sure their lab findings actually held true in the real world. So they used some powerful computer-based analyses to look at data from several large studies of men with prostate cancer.
So bringing the lab and the clinic together.
Exactly. And they look at men at different stages of the disease.
Okay, so after all that work, what did they actually find out about this TBX2 protein? Like, what's the big headline here?
The main takeaway is that they discovered that TBX2 acts as a sort of molecular switch in castrate-resistant prostate cancer. It basically flips the main signal that's driving the cancer's growth from the androgen receptor to a different receptor called the glucocorticoid receptor.
Glucocorticoid receptor, that's a new one.
It is, but think of it like this. The cancer cells have this preferred fuel source.
Right, the androgen receptor.
Right. But when that gets blocked, they need to find a new source of energy.
So the glucocorticoid receptor is like their backup generator.
Exactly. And TBX2 is the one who flips the switch.
Okay. That's a great way to visualize it. So what's the normal role of this glucocorticoid receptor in the body? And how is TBX2 messing with it in prostate cancer?
So the glucocorticoid receptor is like a cousin of the androgen receptor.
Okay.
It also responds to hormones, but these are hormones like cortisol.
Cortisol, that's the stress hormone, right?
That's the one. It's involved in our body's response to stress and helps regulating things like inflammation.
Got it.
Now here's what they found. TDX2 actually directly reduces the production of the androgen receptor.
So it's like it's sabotaging the primary fuel source.
Exactly. It does this in a couple of ways. It physically attaches to the control region of the androgen receptor gene, sort of putting a roadblock in the way.
Okay.
And it also reduces the levels of another protein called GATA2, which usually helps the androgen receptor do its job.
So it's a double whammy.
Yep. TBX2 is actively working to turn down the androgen receptor signal.
So it's putting the brakes on the androgen receptor?
Yep.
But what about the glucocorticoid receptor? What's TBX2 doing there?
Well, on the flip side, TBX2 is actually increasing the production of the glucocorticoid receptor.
Really?
Yeah, it's like it's setting on the gas pedal. It does this in a couple of ways as well. It directly binds to the glucocorticoid receptor gene, promoting its activity.
Okay.
And remember those protein partnerships we talked about?
The dance party.
Right. TBX2 physically interacts with the glucocorticoid receptor protein, giving it a boost.
So let me get this straight. In prostate cancer that's resistant to treatment, TDX2 is basically turning down the signal from the androgen receptor and simultaneously turning up the signal from the glucocorticoid receptor.
Yes.
And this switch is what allows the cancer to outsmart drugs like enzalutamide.
You got it.
Huh.
By shifting its reliance from the androgen receptor to the glucocorticoid receptor, the cancer can keep growing even when enzalutamide is trying to block the androgen receptor.
So it's like a bait and switch?
Pretty much. The cancer is pulling a fast one on the treatment.
And the research didn't stop there, right? They also identified a potential way to interfere with the switch.
They did. They found that if you can target the protein complexes that TBX2 is involved in, you might be able to disrupt this whole process.
Interesting. And all of this wasn't just happening in the lab, right? They saw evidence of this in samples from real patients.
This is the really important part. They found consistent patterns in actual tissue samples from men with castrate-resistant prostate cancer. Higher TBX2 activity was linked to lower androgen receptor activity and higher glucocorticoid receptor activity, just like they saw in the cells.
So it's matching up.
Yes, exactly.
But here's the really interesting part. They also saw similar trends in samples from patients who were in earlier stages of the disease, men who initially responded to treatment.
You're right. That's really key.
So this switch might be happening even before the cancer becomes fully resistant.
That's what it suggests.
Yeah.
And that could have huge implications for how we approach treatment in the future.
Okay. Let's talk about those implications. How could these findings change how we think about and treat prostate cancer?
Well, first off, because they're seeing these relationships even in early stage disease, it means we might be able to use these proteins, TBX2, androgen receptor, and glucocorticoid receptor, as markers.
Okay.
We might be able to identify which patients are more likely to develop resistance down the road.
That would be amazing.
They would. We could potentially tailor their treatment approach right from the get-go.
That sounds like a big step towards personalized medicine. What about new treatments? Does this give us any ideas for how to actually combat this resistance once it develops?
Definitely. This research opens up a whole new avenue for treatment possibilities.
Okay.
Now, we know this switch from androgen receptor signaling to glucocorticoid receptor signaling, driven by TBX2, is a major player in resistance.
Right.
So that means therapies that aim to disrupt this switch to stop it in its tracks could be really effective.
I see.
And one way to do that could be to develop drugs that specifically target the activity of TBX2 itself.
Okay. Now, I remember reading somewhere that directly targeting the glucocorticoid receptor can cause some pretty nasty side effects.
Yeah, you're right.
So does this new research, this focus on TBX2, offer a safer way to go after this resistance mechanism?
That's a really important point, and you're absolutely right.
Okay.
Blocking the glucocorticoid receptor directly throughout the whole body can have a lot of side effects.
It's a pretty important receptor.
It is. It plays roles in a lot of different processes.
Yeah. Right?
But what's exciting about this research is that it suggests we might be able to indirectly target this switch specifically in the cancer cells.
Okay.
We could interfere with TBX2's activity or its interactions with other proteins just in the tumor.
So it's like a more targeted approach.
Exactly. We can hopefully disrupt the glucocorticoid receptor signaling just where it's causing problems without affecting the whole body.
And you said earlier the fact that they saw these patterns in actual patient data gives this whole thing more weight?
Well, absolutely. It's not just something happening in a petri dish.
Yeah.
The fact that those lab findings are reflected in multiple groups of patients, that really supports the idea that these mechanisms are actually important in real-life prostate cancer.
So it's more likely to translate into something that can actually help people.
Yeah, exactly.
Now, the study also mentioned a specific drug called SP2509. What was that all about?
SP2509, it's a drug that inhibits a protein called LSD1.
LSD1. Okay.
And, this is where it gets really interesting, they found that LSD1 actually interacts with TBX2.
Another one for the dance party.
Right. And even more exciting, they found that SP2509 could not only disrupt the TBX2 LSD1 interaction, but also the direct interaction between TBX2 and the glucocorticoid receptor.
Whoa, so it's a double disruptor.
Exactly. It can mess up TBX2's interactions on two fronts. And that suggests that drugs that target this pathway, they might be able to indirectly inhibit the glucocorticoid receptor signaling and potentially resensitize the cancer cells to enzalutamide.
So enzalutamide might start working again?
That's the hope. And it's worth noting that similar LSD1 inhibitors are already being tested in clinical trials for other types of cancers.
Interesting. So there's already some groundwork laid.
Exactly.
Of course, no research is perfect. There are always limitations and things we still need to figure out. What did the researchers highlight as next steps?
So they acknowledge that while their models and patient data are really encouraging,
Right.
the next big challenge is to develop even better, more sophisticated models of castrate-resistant prostate cancer. They need to really dig deeper into all the molecular players involved in this whole androgen receptor to glucocorticoid receptor switch.
Because there are probably other things going on that we don't even know about yet.
Exactly. There are likely other factors that are contributing to resistance that we still need to uncover. It's a complex process.
And taking these lab findings and turning them into actual treatments that can help people, that's a whole other hurdle.
Oh, definitely.
Yeah.
While SP2509 showed some promise in the lab, we're still a ways away from having a drug that can effectively and safely target this mechanism in men with prostate cancer.
So more research is needed.
Definitely. It's a long road from the lab to the clinic. But this research lays a really important foundation.
And it's important to remember that cancer is incredibly complex. This isn't the only way resistance develops, right?
Absolutely. Prostate cancer resistance is a multi-headed beast.
Okay.
This study has illuminated one important piece of the puzzle, but there are likely many other ways that cancer cells can evade treatment that we still need to understand.
So this is just one piece of the puzzle, but an important one.
Absolutely.
Now, to give our listeners a sense of what the researchers themselves are saying about this, I want to share a quote from Dr. Nandana, which was included in that Health News article.
Oh, that's a good idea.
What did he emphasize about the significance of this work?
Well, Dr. Nandana really stressed that developing resistance to these androgen receptor-targeted therapies remains a major roadblock in successfully treating advanced prostate cancer.
It's a huge problem.
It is. And he really highlighted that this study offers a potential new way to both predict which patients might develop this resistance
Okay.
and then figure out ways to potentially overcome it. And the key, he says, is understanding the interplay between TBX2, the androgen receptor, and the glucocorticoid receptor.
So looking at how all three of these players interact.
Exactly.
And I think he also mentioned the excitement around the possibility of making those existing drugs effective again.
He did. He talked about how scientists have been working hard to find ways to resensitize prostate cancer cells to androgen receptor signaling inhibitors.
Right.
And this research could be a major breakthrough in that effort.
And he seemed particularly optimistic about targeting TBX2, right?
Yes. He underscored the potential advantage of targeting TBX2 specifically because it might allow us to block this harmful switch, this shift to the glucocorticoid receptor, without the risk of severe side effects that could come with directly targeting the glucocorticoid receptor.
That's a really important point. So it's like a more precise and hopefully safer approach.
That's the idea.
Well, this has been an incredibly informative deep dive. We've learned a lot about how prostate cancer becomes resistant to treatment
Yeah.
and some exciting new avenues for potentially overcoming that resistance. So, as we wrap things up, what's one final thought you'd like to leave our listeners with?
Well, thinking about this study, how prostate cancer cells have evolved, this clever way to escape treatment, it really makes you wonder how many other similar escape routes might exist in other types of cancer.
Hmm, that's a really good question.
Right. And if we could identify and understand these mechanisms in other cancers,
Yeah.
imagine the possibilities for developing more durable, more effective therapies in the future. We could really start moving toward a more personalized approach to treating cancer.
It's almost like if we can understand how cancer cheats, we can develop better strategies to outsmart it.
Exactly.
So for our listeners who are interested in learning more about this research, maybe keeping up with future developments, where would you suggest they look?
Well, I would definitely encourage them to stay tuned for updates on clinical trials involving those LSD1 inhibitors
Okay.
and any new research specifically on the role of TBX2 in prostate cancer.
So stay curious.
Yes. And for reliable information about ongoing research efforts in prostate cancer, organizations like the American Cancer Society are a great resource.
Fantastic. Well, thank you so much for joining us for this deep dive today. It's been a fascinating look into the ongoing fight against prostate cancer.
My pleasure. Thanks for having me.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.